Good morning, everybody. It's very early for a lot of you, but I'm also a teacher, and I know that the best students are always, on time, sitting there with scattering eyes, and only let's say, the bad students come late because they went out the evening before and they don't have time to get up. So it's a pleasure to be here. Thanks to Boehringer, they gave me some time to talk about the most interesting thing for me on the world, that's pathogenesis of viruses. And the most difficult pathogenesis are the pathogenesis of viruses that are causing reproductive failure. And I've always been interested, and for me it's a pleasure also to have two strong scientists on my side. They're two females, they're really damn good, and I'm following them already for a long time, and I'm convinced in the future they will keep on going. So even when I'm dead, I'm convinced pathogenesis will stay alive for a long time. So what I will try to do is it's difficult to talk about mechanisms, but I will try to make it easy for veterinarians, so at the end that you will understand how to approach reproductive failure as a vet. And this is extremely important. It's easy, but you have to use it. And most people don't use that system. And I will show you, and I hope that you will use that system. It's very normal, it's very logical. And I know that a lot of companies like to make it extremely complicated because, then it sells vaccines. But for me, I'm a vet, and I really want to have the diagnosis correctly. And before you go into prevention and curing things, you should have a good diagnosis. Next one. So if we talk about the normal gestation in a sow, then you need two things. That is a hormonal control, and as a student that was the only thing we got in the past, the progesterone. So that thing is just keeping your uterus calm. If your uterus would not be calm, you would have an abortion because the muscles in your uterus would be active and throw the thing out. So this is the only thing that thing is doing. This is the hormonal control, and then beside the hormonal control we have also the immune responses. So you should know that a pregnancy, I call it always a tumor, because that thing is antigenically not normal. That should be rejected, and it's growing, it's proliferating. So the immunity, a normal immune response would say get the thing out, but it's not happening. So it is a pure immune suppression from the fetus, telling the uterus, you don't reject me. So there is a very important, they call it immunological control, and there should be a crosstalk in between the fetus and the mother. And this is a very intriguing topic. My wife is in reproduction and infertility, and I always tell her, you should work more on that. But nobody wants to do it. But this is the most interesting thing to do. That is the crosstalk between leukocytes on both sides, leukocytes of the fetal and the mother side, and there should be a crosstalk in between them. And very important in this at this point is that they call it microchimerism. So that means that there is some crossing of cells from the mother to the fetus and from the fetus to the mother. This has been disputed for a long time, but now it's generally accepted that there is a clear crosstalk in between the two individuals, being the mother and the fetus. This is also very important. So there is a nice crosstalk, and the crosstalk says stay in. So the fetus stays into the uterus just because the whole system says, calm down, don't react to those foreign antigens. Then how do viruses cause reproductive failure? And I want to keep it simple. So there's a very big, I think the biggest group are just causing general disease. This is just, due to, as you can see, pro-inflammatory cytokines causing distortion of cytokine balances. So beautiful examples is swine influenza. You know that if there's a storm of influenza coming over a farm, sows get fever, and then you will have for sure abortion. We will come back to that later on if we talk about diagnosis. For me, I call this top boring. People that work with influenza, it's a superficial infection. It's just infection of epithelial cells, and that's it. And let's say the reproductive failure has nothing to do, let's say, the pathogenesis. It's just a kind of a fever reaction and causing reproductive failure. The most interesting part is the bottom line. The bottom, that's the replication of pathogens in the reproductive tract, embryo and fetuses. That's more interesting to me because the question is, how is a virus getting there? So it should do efforts to get there, to do the thing, and to destroy things, and that's very important. And that's why I'm very interested in that. And they do that by just damaging the placenta and or cytokine balance. They can do that on the spot. And of course, they can also do that by direct negative impacts on embryo fetuses. So if they kill embryos and fetuses, of course, they will also cause problems. We have two types of viruses that do that. We have DNA viruses, RNA viruses. And in the DNA viruses, interesting ones are Ojevsky disease virus. This thing is gone in Europe, which is a pity because I like that virus. Parvoviruses, I think this will be the topic of today. PCV2, I think Tanya will go into depth in that. RNA viruses, PRRS virus, enteroviruses, encephalomyocarditis virus, and classical swine fever virus. Of course, with African swine fever coming to our place, we should in the future also add that one to the DNA viruses. First of all, let's keep it basic. So how can a virus get into the uterus? I would say, pour it into the uterus. This is by contaminated sperm, of course. This is the fastest way to get into the uterus, as this is the moment where the cervix is open and that the things can get into the uterus. So this is easy for the virus. And if you look into that, then you should understand that there should be, first of all, virus in the semen, and then it should be introduced in the sow, and then it should survive in the sow. So there are a lot of conditions before the virus can stay there and do the thing. So you see here, and there's a lot of, if you go in literature, a very good correlation between virus excretion in sperm and viremia in that animal, which is logical. If there is no virus in the blood, it will not enter organs that are at remote places. So this is extremely important. So we have here DNA viruses, Ojevsky's, Parvo, PCV2, and you can see here the periods during which they can be secreted by the boar. RNA viruses too, with PRRS, enteros, and classical swine fever. So you can see that the virus is shed in sperm for quite some time. When the virus gets into the sow, then it can, as you can see here, have an impact on the embryo. For instance, Ojevsky's Parvo PCV2 virus replication and death of the embryo at an early stage, causing, of course, repeat breeding because you get resorption. RNA viruses, PRSV, enteroclassical swine fever, you have for PRRS, and that's important, no virus replication. You will get, and that's always the point, in the field you get sometimes embryos at the back of the sow, and then they, from the very beginning, when PRRS emerged, they were telling me, Hans, there's virus in it, you should find it. There's no virus in it. The virus is on the other side, it's in the placenta, destroying the implantation region, destroying the macrophages that are important for implantation, but they are not inside the embryos. So this was so the virus is not replicating in the embryos at a very early stage, and the reason for that is very easy. There are no receptors at that moment. So you start with embryos. They are just a mass of cells. There are not a lot of differentiation markers on those cells, so they're not susceptible for a lot of viruses. Enteros and classical swine fever can replicate and also cause death. So we have a lot of viruses causing repeat breeding in the sows. This is also important. In the past, people were busy with looking at what stage the embryos can be infected. And some of them are not studied very well, but most of them, as you can see, they will not infect the embryos at an early stage. So you have 2 to 8 cells, morula blastocysts. So this is the first week. This embryo is fully protected. The reason for that is very simple. This is a beautiful area. This is the zona pellucida. This is really, these are sugars. This is a network of filaments, and this is causing a very good barrier against viruses. This is a typical example of coevolution. So there's a Pathogens want to attack, of course, but the organ says we will protect. So the zona pellucida is one of the most beautiful examples of barriers against pathogens. However, if they hatch, so if your embryo gets out of the zona pellucida, they become fully susceptible, and that's from 7 days. And then you can see, this is for instance PCV2, that a lot of viruses start to replicate in that embryo. You have here one, and that's PERS, of course, will not do the job. And the reason for that is there are no receptors yet. So that thing has to wait, especially there we need macrophages to come into that embryo, and that's not the moment that macrophages are produced. So this is too early for PERS. That's also why when you have PERS in the uterus in a very early stage, you don't have killing of the embryos, but you have infection, of course, of the sow. This is the beautiful zona pellucida. I had a crazy person, Geert van Roosen. He's now even a vet here in, I think, somewhere in France. I forgot where he was. But he was studying these holes. He was, he was taking EM pictures, and then he was measuring all those holes by hand. I told him he was crazy. I said just use beads with different sizes and have look how deep the beads are getting into the zona pellucida. He did both, which shows that he was a very good PhD student. And he came to the conclusion that these holes are 30 nanometer. So that means 30 nanometer, this is extremely small. That means a lot of viruses like Ojeski disease virus, PERS, and classical swine fever virus, they will never enter the zona. But of course, if I told you before that, as you can see, that also here the very small one, enteros and parvo, they will not infect the embryos. There should be another reason, because if you inject, so if you take a needle and you inject the virus, you can infect the embryo. So the zona pellucida is really a barrier. And the reason for that, it's not only a physical barrier, but also one of the most beautiful chemical barriers. It is sticky. And this is not only true for the zona pellucida, but it's the same for the mucus. The respiratory mucus forms also a barrier. Also big holes, but also sticky. And it is also the same for intestinal mucus. So the mammals made a lot of sugar barriers against viruses. This is just to show, this is parvo, for instance. This is already an elongated embryo, and this one is infected, and you can see that one is, of course, totally dead. We did in the past some work on PCV2. I will not go in detail, but we just collected. This was a huge job. Took 2 years of work of one student collecting embryos, inoculating or not inoculated, putting it back into receptor gills, then euthanize them 14 days later, and have a look. Then we were flushing out the embryos, and we were looking, to those embryos to see. And we can be short. We saw that in the mock there was nothing going wrong. In the inoculated with embryos inoculated with PCV2, we saw some. We didn't find embryos back. That means that you had already resorption. Some of them we found some embryos back, but we saw that they were fully infected. And you could clearly see when we made some sections, you could see the infected cells inside the embryos. This work that we did on PCV2 demonstrated clearly that this virus belongs to the SMEDI viruses, just like enteroviruses and also parvoviruses. So this is, let's say, getting into a sow from the back, by contaminated semen. Let's now go to the front of the animal. So infect a sow from, oral or intranasal, whatever. So then you need, of course, if you want to go to the uterus, you need a trick to go into the blood and to end up in the uterus. And there are a lot of viruses doing that. So RNA virus, PERS, of course, classical swine fever, enteroviruses, and everybody forgets always the enteroviruses. Encephalomyocarditis virus, which is exceptional because this thing is coming from rodents. And if you have a nice control of rodents, you will not see the encephalomyocarditis virus. And very important is that you have to separate those viruses in viruses that go to the uterus in leukocytes. This is cell-associated, I call it, or you have viruses that get over the blood, but in a free stage. So that I call that cell-free. And I classify them as cell-free, cell-associated, and the story will be into 2 directions. So the first one is the cell-free. PERS is a beautiful example. The others, this has been mentioned as virus being cell-free, but I have some doubts. I think some people should investigate parvo, for instance. I think there are a lot of things to reconsider with parvovirus. I think I will not say we got it wrong, but there are a lot of things to discover on parvovirus. So I really would activate young people. I'm getting old, but the young people in the audience to stimulate research again on parvoviruses. For PERS, it's simple. This thing, as you can see, these are the blood vessels. In blue, the maternal side. In pink, the fetal side. So what you need is this free virus has to go to the fetal side. I remember discussions with my teacher, Professor Pensart, a big guy. He always said, Hans, this is simple. You get holes, during gestation, the thing becomes so, stretched that you get holes, which is stupid. There are no holes in animals, it's all fully controlled. So there should be a way how the virus gets in there. So there is no way that this thing will go just as a virus and say, I will have today the fun of my life, and I will just cross a placenta as such. That's not possible. So there should be a way. And for PERS, this is a very beautiful story. For PERS, this virus is just, as you can see here, in a free stage. In the blood, you have monocytes also down there. And monocytes, especially at the end of gestation, the monocyte becomes a macrophage. When the monocyte sticks and leaves the blood vessels to enter the placenta, the maternal placenta, and this is happening late in gestation, the reason for that is simple. You will get at the end a rejection. So parturition is nothing more than a rejection of a tumor. And the tumor are fetuses, so the mother says, get out. And she creates a kind of an environment from immunosuppression into immunity. And this immunity has to be controlled, of course, by cells. And the placenta has to react against the fetuses. And who's the biggest director? Monocytes, macrophages, always. So they control the whole response. So what you have here is the macrophages are just leaving the blood vessels. They have the receptors there, and the virus is just infecting. And that's the beauty on the whole system. The macrophages become infected. These are just blood vessels, and you can see here beautifully inside the blood vessels. And this is not exceptional, because you can say people are just looking for hours to find those cells. It's full. If you have a placenta, you find these cells everywhere in those blood vessels. Then, of course, you get out, you're infected, and you start to infect an environment. Of course, if you infect the environment, then you're still at the maternal side. So you have to end up in the fetal side. And then this was a big discussion. I had a very strong Belarusian student, Uladzimir. He was brilliant. And I told him, how should we look into that? If you say that the cell is just leaving from the mother side into the fetal side, we should find in male fetuses, we should find female cells. And that's what he did. And he found quite a lot, as you can see here. So red is Y, green is X. So you have here a female cell. This is XX. So that means in this male fetus, you have cells from the mother, which is at that moment when he published that, that's already maybe 10 years ago, because I'm getting, growing old is going fast. But this was beautiful. I think nobody reads that paper, but it's a brilliant paper showing, of course, this is, for me, it looks like a macrophage, but you have to check it, and it's very difficult to do in situ hybridization in combination with detecting antigens. But this is really beautiful work, and also very interesting, because this shows also the crosstalk between the mother and the fetus, and also the opposite, the fetus and the mother, which is a huge science to do in the future, because this will also have some practical things in direction of human reproduction. So this is, I make here a macrophage, that this is just leaving the mother side into the fetal side. We don't have proofs for that, but I'm almost, let's say, 99.9999% sure that this is happening. Then the next thing is that we get a full-blown infection of everything. So this is the mother side, the fetal side, and this is what you see. So this is the mother side, the fetal side, and you have positive cells, macrophages, all over there causing the problems. And this is for PERS. This is also, but I will not go into that. I think Andrea will go into detail. There is a very interesting thing going on with the immune response there in the placenta. Then later on, of course, the thing is just spreading, and you get this destruction for PERS. This goes in the, this is physiological. This is apoptosis. This PERS is a soft killer. It's not putting the things in necrosis, which is very smart. So it's killing in a soft way. But of course, killing is killing. And if the virus is continuing killing, you get a lot of apoptosis in all those areas, not only of the infected cells, but also of bystander cells. You get a lot of destruction. And if you have a lot of destruction, then you will have also a lot of, as you can see here, a lot of lesions. And this is also work from Oladzimir. He clearly demonstrated that if you have lesions, you will clearly, I'll have to push this one. It's not working extremely fast, but okay, here it is. This is a normal placenta, so this is the fetal placenta, this is the endometrium, and you have a full contact of those two layers. If you have PERS, you get a lot of, destruction of that area, causing a lot of lesions, and this is what you see, and that's also always what I say. You don't get killing of a fetus, you just destroy your fetus because your placenta is destroyed. And I think Andrea will come back to that later on. So PERS, I don't have to present the clinical signs. You live with PERS, so you know what it is. Then we go in the direction of the next one. This is let's say the cell-associated viruses. For my PhD, I worked with the Ojeski disease virus, which was a brilliant virus, and I did a lot of work on that. I remember I started my PhD with Pensart, and he said, Hans, we have abortions in vaccinated sows. Solve that. That was my project. So if we now have a PhD student, we always have to write down 20 pages to stimulate a person to do the research. In our time, it was just you got a topic and just do it, which was quite big fun because I had no control. You could do whatever you want to, and I did a lot of vet stuff to demonstrate that, and especially first of all, we demonstrate that monocytes, because monocytes, macrophages are part of my life nowadays. When I go into pathogenesis, and it is a general disease, monocytes, macrophages are always playing a kind of a central role. And that was also for Ojeski disease, that was the game. So the infected cells in the blood, they were infected monocytes. They were the cells that were carrying the virus from the upper respiratory tract to the uterus. That's how the virus got into the uterus. And we demonstrate also, that's also a long time ago, a lot of papers. Nobody read it and will not read it. Let's say mechanistic things, not a lot of people are, interested to read those things, but we demonstrate that the monocytes are just escaping from antibodies in the blood circulation. So even when a lot of people vaccinate at that moment, I will not use names of vaccines and companies now, but a lot of people used inactivated vaccines at that moment, and we knew that those vaccines, they were good, but they were not that good because they could not protect the uterus. The monocytes could still spread up to the uterus and still could replicate there and cause abortions. So the virus there is just in the monocyte and is just doing its thing. So it first will stick to the vessels. Then after, when it sticks, then it will cause a beautiful lesion, as you can see here. And a very important, everything what is brown here is infected. So you get the thrombus in the blood vessels. That's too fast. You get there's a certain delay on the computer, but okay. Yeah, so this is a blood vessel. Here you can see the thrombus, and you can see the virus is just spreading from cell to cell. This is typical for herpes viruses. If you have a blister on your lip, the virus is just, it's creeping. It's going from, that's why herpine it's Greek for creeping, getting from one cell to another. And this is beautiful. You can see it here. The virus is going from one cell to another, and is just invading the tissue, first the maternal tissue, but then later on it's going further and further and ending up into the fetal site, in the pink site. And that is, so PERS is, for instance, is reaching the fetus is in a total different way than, for instance, Ojeski disease virus. And this is also beautiful to have looked at that. So this is what. So nowadays, Uladzimir was successful in keeping the placenta together, so the endometrium and the fetal placenta. And Andrea, you know that this is not very easy to do that. So this was clumsy. This was my work. So I got they got loose, but you can see here a replication on the tip, and we can find here on the other side with the microscope you could see already 3 or 4 positive cells. This was I killed the sows at that moment. It was easy to kill sows. I killed the sow early after I was inoculating the cells, and this was what we got. So this is a clear proof that the cell-associated spread is important for Ojeski disease virus. And then, of course, if you have this, then you can, Ojeski disease virus is just spreading all over, and there's no control. Once in one cell, it's getting all over. So you cannot stop it. And that's why Ojeski disease virus was one of the most important causes of abortion. There was no way that you could control it. And that's not only for herpes virus infections in sows, it is correct in all species. So herpes viruses are successful. I have to move on. They're already do like this to me. So so this is what we get then. This is abortion. Very important is the interfetal viral spread. So this is PCV2, parvo, entero, and PERS, and Ojeski disease and classical swine fever. So the last two are very strong in spreading over the uterus. PCV2 is a loser. That one has a lot of problems to cross from one fetus to another. So this depends on viruses. The clinical outcome for fetuses, so if you have infection during the different days of gestation, so a lot of viruses are just killing the fetus in the very beginning, as you can see. And there are a lot of viruses that are killing all over. Ojeski disease virus, sometimes also classical swine fever virus. This depends on the strain that you're using, because there are a lot of strain differences. And very important is from day 70, the fetus can start to react. So a fetus can react in an immunological way and can induce immune responses, and at the end you can have some fetuses that stay alive. Enteroviruses, parvo, PCV2, when they get infected at the end of gestation, they can already react immunologically and the fetus can survive. Ojeski's, classical swine fever, most of them will die at the end. For the sow, Ojeski disease virus, abortion all over. PPV, PCV2, and enteros give SMEDI syndrome, so you get mummification, stillbirth. Those viruses are not causing abortion. That's always the same. People come with bags of fetuses to me, and then they say, Hans, it's parvo. Then I say, come on, guy. This is maybe exceptionally you can have one sow, but this is not. The general picture is parvo, is abortion. This is not due to those SMEDI viruses. This does not. There's no A of abortion in SMEDI viruses, because people that want to reproduce, because this is the most important thing, that is the postulates of Koch. If you want to prove something, you have to reproduce it. And everybody who's working with parvo and is inoculating the virus in the pigs gets a SMEDI syndrome. So SMEDI is SMEDI and is not abortion. PERS is typical different. That's late abortion, early farrowing. Very typical. So also there you can have a clear print. And that's it. This is very important for if we go to the and I will go straight on that because I have to move on. This is all not important. But these are all just simple pictures. But this is my 3 last slides to show what is now important of the story that I made. Very important is you have to start with the main complaint as a vet. And there it all goes wrong. You don't think. When you have reproductive failure, you grab things and you throw it to people. You have to think first, because you can already make a decision. So if you have a main complaint, abortion, then you should know that, and it is all over, different stages of gestation. So you should know that most of those things are just caused by fetuses. When there are fetuses, also very important, if you have fetuses and they're very beautiful, I call them, you can eat them if you see, they're so beautiful. They just, the sow decided to get rid of those fetuses. So when they're out and you look to those fetuses, they're even in, as you can see, I don't know how you, rigor mortis, this is Latin, of course. I hope everybody understands this. They're firm, they're in rigor mortis. That means they were alive when they were born. There is no pathology. So you don't have to, and everybody is looking to the fetus. Damn, the fetus are okay. You should look to the sow. And that is what is going wrong. So the sow is normally, in this case, feverish. So if you take the temperature of that animal, you will find out. And this is typical for influenza virus and some other pathogens that are causing fever. So this is the main cause of abortion. If you really have abortion and there is pathology, as you can see here, no fresh, partially mummified Ojeski disease virus, you don't have it anymore. Classical swine fever virus, you don't want to have it. So these are, you don't have those. You're lucky, you don't have those things anymore. If you're working in Asia, you will still see those things. And these are just, Ojeski disease virus and classical swine fever virus. If you have SMEDI, and this is very, this is stillbirth, mummification, embryonic death, and infertility. These are clear pictures. That means the problems are at the end of the gestation, when the fetus is coming out. That this has nothing to do with abortion. Then you have to think on the SMEDI viruses. And that's also not a big problem with parvo, PCV2, entero, and encephalomyocarditis virus. So this is not difficult. But I know a lot of people will start here to discuss, yeah, maybe, there's something, a lot of people start to think on PERS here, which is, you can sometimes have some starting mummification, but real mummies you will not have. So then that's not correct. So here in this, if you have SMEDI, virus isolation, PCR is very easy. Antibody detection is good, but you have to be careful. I don't want to use any more antibody detection. This is crappy. We published one paper in Virologica Sinica, just to show that if you have antibodies in the fetuses, it can come from sows, because in sows you can have antibody titers. In Belgium, 160,000, in NIPMA, 500,000. This is so huge that little leakage of whatever will end up with antibodies in the fetus. So this is very dangerous to use antibodies to prove your thing that the things are infected with the virus. And then, so we had already abortion, SMEDI, and the third is just the PERS. PERS is just causing late abortion, early farrowing. I think everybody knows it. If you see it, you don't have to think twice. And then very important is, if you want to do a diagnosis there, PCR is the technique of preference, of course, because viral isolation, the biggest problem with PERS is it's not stable in the outer world. It's very difficult to find a virus in those fetuses. And then you make a decision, this is PERS. So I hope these 3 slides are extremely important for you as vets. Don't mess it up. This is very logical. And very important is, make a general thinking. So don't go to one cell, but think, what is the problem on the farm? This is very important, so that you make the correct diagnosis. I oversimplified it, but sometimes that's good, because if you can make people completely mad with very detailed things, and then you lose control of your diagnosis, and don't do that. And this is it. Good, eh? You were scared that I would not stop in time, eh? But I'm getting older, so I can stop everywhere, so no problem. Thank you for your attention, and I think there's time for a few questions. Thank you. Thank you. Thank you for your